Okay. Good morning, everyone. Welcome to the second symposium on big data algorithms for biology. So this is the second year that we are repeating this event. I welcome you all. And I should mention that the highlight of the last event was the engaging questions asked by students after each talk. So I hope to see the same energy from students this year also. I request, so we were waiting for our dean. I request Professor Navakant Bhatt to please come and share his opening remarks.
Very good morning to all of you. Sorry, I got a little late here. Thanks, Professor Chirag and Professor Debnath, for organizing this symposium. Just catching my breath. So I think the theme of this symposium is apt, big data and algorithms for biology. As you know, it's often said that biology is the next frontier for this century, right? Sort of unraveling, various mechanisms and, getting the deeper understanding thereof. I think in this context, it's pertinent to say that the convergence of, all the information technology revolution that we have had over the last few decades, and the new excitement about artificial intelligence, machine learning, natural language models, and so on and so forth, and their intersection on various fields within biology, I think will have a far-reaching impact in the years to come. And the Institute is seized of this matter, and it has embarked on many new initiatives to address this new frontier. And this symposium is, part of that many initiatives that I was talking about. One of the very recent initiative, if I may mention here, which of course Professor Debnath is also very much involved in that, is AI Center of Excellence for Health, which is a multi-institutional initiative led by Indian Institute of Science with many research institutes as well as industries participating in it. So I think the time is opportune, in my opinion, and I sincerely hope that the practicing scientists within these two seemingly diverse areas, that is biology on one hand and data sciences on the other hand, coming together will really herald many new insights in the way life is, and thereby also offer many solutions, be it healthcare or otherwise, right? So I hope that you will have a very rewarding information exchange during the rest of the symposium. And again, congratulate Professor Chirag and Debnath for initiating this. Thank you, and hope to have some interaction with you later. Thank you.
Thank you, Professor Bhatt. Welcome all participants and our esteemed speakers. So, point to note, please keep your mobiles on silent mode. Next point, our other guest, Professor Gopalakrishnan, he is stuck in traffic and he couldn't make it, so he was the other guest, who actually is the convener of the Axis Bank Center who is funding the symposium, so his presence was also expected, but he's stuck in the traffic. So we'll move on to our first distinguished lecture by Professor Partha P. Majumder. I will give a brief introduction. He needs no introduction. He's well known across the country and the world. He's among the top scientists in the world in the area of population genomics. Currently, he's the National Science Chair of the Government of India. He holds adjunct positions in several institutes, including IISER Kolkata. He is known for the rice genome, where he participated. He has, he's known for the rice genome, in which he participated and contributed from India. He leads many other initiatives in India, and he's very popular among students in genomics and genetics. He's originally a statistician and has worked extensively in anthropology. I'll not take much time and request Professor Majumder to take the stage. Thank you.
Everyone, I'm actually quite overwhelmed, primarily by the affection shown by the organizers. I do have a background in statistics, but I've moved along and done genetics most of my life. I generate big data, I analyze big data, but I don't develop algorithms per se. Do develop some statistical methods for handling big data, but really, this is not the forum where I should be talking. But again, like I said, that the affection shown by the organizers in having me give a talk, I'm really overwhelmed by that. I'm going to talk about human history and genomes that we use for making inferences about human history. And I'm also going to talk about a little bit of culture as it spreads and how genes and culture co-evolve. This is going to be primarily a story about human evolution. The story uses tools of archaeology, which I do not understand, so I'm not even going to get into the technical details of that. And then it uses tools of genomics and genetics for making these inferences, and I will not get into the technical details of it, but I probably will hand wave some of the paradigms that we use without showing you the gory details of the mathematics that underlies, mathematics and statistics that underlies the inference. So you will have to excuse me for not being technical and being only a storyteller, but that's what I've told the organizers I'm going to do, and they were okay with it. So I'm happy to be here. So of course, we have to start with Charles Darwin when we talk about human evolution, and he wrote this book on the origin of species. And in that particular book, he made no mention of the humans, or he essentially wrote this one line: light will be thrown on the origin of man and his history. That's all he wrote in this book. But he did keep his word. A few years later, in 1871, he wrote this book exclusively devoted to humans, The Descent of Man and Selection in Relation to Sex. And then he said, he made this point that man and great apes have a common ancestor. And today, from various angles, however we look at it, there is no doubt that man and great apes have a common ancestor. But the kind of evidence that he provided will look frivolous to most of us. Of course, the first one is okay in the sense of bodily structure, tissues being similar, etc. Common parasites, I mean, it might look a little frivolous. So anyway, he provided these kinds of evidence in order to argue that man and great apes have a common ancestor. So I'm going to tell you a story, like I said, that it's going to be a story. I'm going to be a storyteller for now. My story comes in two parts. The first part begins about 5 million years ago and ends about 130,000 years ago. When I talk about these dates, like 130,000 years, 5 million years, etc., all of these dates you have to take it with a large pinch of salt, because these are estimated dates and have large standard deviation oftentimes. I will mention some of those as I go along. The second part of my story will begin around 130,000 years and continues to the present day. The part one of the story, like I said, it begins about 5 million years ago. And what happened 5 million years ago was a population of African apes split into two distinct species. One line gave rise to the hominids, eventually leading to the humans. The other line continues, and we have chimpanzees, bonobos, and so on and so forth. I'm also going to jump in time, at least the first part of the story, very rapidly. So 1 million years have jumped, and 4 million years ago, one of the species on the evolutionary path to humans began spending most of its time on 2 feet, and that was the genus Australopithecus. There are various species in Australopithecus, and the species that we are most familiar with is called Australopithecus afarensis. Australopithecus afarensis was dug out in Ethiopia, and the discoverer of the fossil was Donald Johanson. This is the reconstruction of the skull, and this is also very famously known as Lucy. This particular specimen is known as Lucy primarily because Johanson was listening to the Beatles song Lucy in the Sky with Diamonds, and that's when he found a bone and eventually decided to call this specimen as Lucy, and we all know this specimen as Lucy. This is Australopithecus afarensis, the first hominid that had a bipedal gait. How do we know it had a bipedal? These guys had bipedal gait. We know because these are footsteps of Australopithecus genus, of members of the Australopithecus genus, that has been preserved on a volcanic ash bed. And I can't read this, but whoever can read these footprints, they will vouch that whoever walked on this volcanic ash bed essentially had an upright gait and was a bipedal. This rock bed has been dated 3.7 million years ago. So, like I said, that about 4 million years ago, Australopithecus, the genus Australopithecus arrived. This was also found by a very famous family, member of a very famous family called the Leakey family, who spent generations digging up fossils and reconstructing human evolution in Africa. This was discovered in 1975 by Mary Leakey. The upright stance that Australopithecus had seems to have set in motion a profound evolutionary change. Hands became free. So they were able to survive better because they could throw stones at predators. They had visibility, greater visibility over the higher underbrush, so they could spot their predators from a larger distance, and therefore their probability of survival increased. And they had dramatically larger brains. We don't know whether this is related to bipedality, but that's an observation that's also correlated with bipedality. Again, I jump another 2 million years, about 2 million years ago, 1.9 million years ago, the genus Homo arose, and the characteristic feature of the genus Homo is that they were able to use tools, stone tools. So, this is the genus Homo, and Homo erectus. There are multiple fossils found in various parts of the world, Africa, Java. This is about 1.8 million years. Once the first species in the genus Homo appeared, it began to spin off new varieties of Homo. And as you can see that there are multiple varieties of Homo, multiple species. We will concentrate, of course, on the Homo sapiens, which is us, and we'll also talk about the Neanderthal man and the Heidelberg man to a certain extent. So we actually arose in Africa, and we came out of Africa at some point of time, started to populate other parts of the world. But when we came out of Africa, and I will provide more evidence that we actually evolved in Africa and came out of Africa, but when we came out of Africa, we saw that there were other kinds of hominids, humans, who were living outside of Africa, and we met them outside of the African continent, and they essentially look like us, as you can see. So, we came out and saw that we were not alone. There were other people who preceded us, and we met them in Eurasia, and they also came out of Africa much before we did. So, if you look at this particular, these are called phylogenetic trees. These are called, these trees have branches. This is exactly a tree. That's the root of the tree, and these are branches of the tree. As you can see that each branch has a name, Floresiensis. This is the famous Indonesian hobbit. Homo erectus, Homo antecessor, etc. The important part that I want to point out is that many of these branches are actually dead. You don't see them any further anymore. Homo erectus branches are essentially dead. And there are some branches that have actually intermingled with a larger branch. So we will talk about the Heidelberg man, Homo heidelbergensis, and essentially the Heidelberg man also looked somewhat like us, quite like us as a matter of fact. And from that particular, large stem arose other branches, and these are two branches that we will talk more about. One's called the Denisovan branch and the other is called the Neanderthal branch. Both of these branches, as you can see, got intermingled with the Homo sapiens branch, and of course, we are widespread throughout the world, and that's this particular branch has lots of sub-branches, and we have completely invaded, expanded throughout the world. So this happened about 400,000 years ago, and about 150,000, 130,000 years ago, we arose in Africa, and then after some time, we moved out of Africa to meet other people in out of Africa continent, and whatever we did, I will mention. So this is where this 130,000 years comes from. It can be 150,000, it can be 130,000. So that's what it is. So that 130,000 years begins our part 2 of my story, which will be a little bit more expansive. So this is what when this is the origins of modern humans. That's about that time. We are less heavily built, we are more mobile, and we have a higher cognitive flexibility. We arose about 130,000 years ago. Again, like I said, I emphasize that don't take these dates as sacrosanct. So this is one particular document, one particular skull that was almost complete, a complete skull, again discovered by a member of the Leakey family, Richard Leakey. So we evolved somewhere in Africa, possibly in the Ethiopia-Kenya region, because of the richness of fossils from that region, dating of the fossils, and so on. We were a small group of people. Our ancestors were hunter-gatherers. They looked like us, high forehead, sharp chin, etc. As you can see that, any of us would mistake that for a Homo sapiens. Today there are over 6 billion of us, and each one of us descended from that small group of anatomically modern humans. Millions of humans have lived and died, but fossilized remains of only a few hundred exist, and therefore that puts a huge burden of speculation if you're going to use fossil data, archaeological evidence, and so on. So that's a huge burden of what else can we do? We can use DNA in order to make draw these inferences about our past. DNA is from the contemporary populations. Today we are able to sequence even ancient DNA, but that only came within the last 15 years or so, or 10 years or so. Otherwise, we use contemporary human populations, analyze their DNA, use statistical methods in order to infer about the past, and I will tell you a little bit of the paradigm. So essentially, if we look at genome sequences of 22 individuals, these individuals are in the present time. These individuals have evolved from the past to the present time. We are using these data in order to infer about the past. What do we do? So essentially, if you look at one generation ago, there were 18 ancestors, primarily because some of these people did not actually leave offspring. They did not have any offspring. Some of them left multiple offspring, as you can see. This process continues, so you have 16 ancestors in the previous generation, and so on and so forth. So this is how it happens. So eventually, this is how the entire relationship among these various DNA sequences will look like. If you remove many of the dead branches meaning individuals who did not leave offspring for the next generation, and therefore in the contemporary population their DNA sequences are not represented. So if you remove those dead branches, this is how the phylogenetic tree will look like. So this is called the most recent common ancestor of these 22 individuals, and this is exactly how the phylogenetic tree would look like. And this whole method of DNA sequencing coalescing in the past is called a coalescent process. The probabilistic framework of the coalescent process was proposed by J.F.K. Kingman. He actually provided the methods of estimation, of methods of inferencing, and so on. This is not an easy paper. It's called the coalescent. It was done by J.F.K. Kingman, and this is what we use. This is one framework that we use. we can now add mutational events. These are DNA sequences that evolved from a most recent common ancestor to these 22 DNA sequences. We can add mutational events, and of course, mutations would have taken place in the DNA sequences. So, we add mutational. Let us say that a mutation arose here. So, if a mutation arose here, all of the descendant DNA sequences would carry that mutation. So, if that was the mutation at that particular position from G to T, then all of these individuals would carry the T mutation. Let us say that another mutation arose here, and that mutation is essentially that particular nucleotide change. All of the descendant DNA sequences would carry that mutation, and so on. So, you could have, even after a mutation has arisen in a particular line of descent, you could have other mutations arising there. So, the process continues, and today what we see is that all of these 22 DNA sequences are not exactly the same. There are mutations in those DNA sequences, and if you look at these DNA sequences, these 22 DNA sequences would have differences. Now, these 22 DNA sequences do not have to belong to the same population. These DNA sequences could have come from different populations, and so let us say that these populations, these DNA sequences come from 3 different populations. The first thing that you see is that there are some particular lineages of the DNA that are specific to a population. There are some that are shared between populations, and if you look at frequencies of the mutations, you will see that the frequencies of the mutations, even when they are shared between populations, may not exactly be the same. There will be differences between sharing of between the frequencies of shared mutations. So, these are our data now comprises this kind of data, where we know that individuals are drawn from specific populations. We sequence their DNA, we find out the characteristic features of the DNA, compare them with DNA sequences drawn from other populations, and then try and reconstruct this entire phylogenetic tree. What we are truly interested in is, when did this set of individuals have this common ancestor? What is the coalescent time to this particular common ancestor? What is the coalescent time to the root of this tree? Those are the kinds of questions that we ask. We can have a framework for that, and the framework of course uses probabilistic techniques, and also uses certain kinds of assumptions. Some of those assumptions may or may not be valid. We now have methods of relaxing some of those assumptions, but again, this is not really a course in population genetics that I am doing, just giving a flavor of what we do. So, we can actually look at the TMRCA, which is time to the most recent common ancestor, which is really the coalescence of all of these events, all of the coalescent events dating back to the most common ancestor. So, this is the first coalescence, this is the second coalescence. So, we can ask the question, after the first coalescent event happened, how long does it take for the second coalescent event to happen? So, under those assumptions, under the probabilistic framework, we can actually estimate those times, and we can actually show that the coalescence time follows an exponential distribution that has a particular mean and a standard deviation. Given that there are J lineages, what is the probability or what is the probability distribution of the next coalescent event, and this is what we can derive. Just to give you an idea as to what the trees will look like, if you have a constant population size, so these are, let us say, individuals drawn from a population, over generations their size remains constant, what you will find is that most of the coalescent events will happen near the bottom of the tree. On the other hand, not all populations will be of constant population size, they will also increase, and if you assume that the populations are exponentially growing, then most of the coalescent events will not happen at the bottom of the tree, will occur at the distinct path. So, even by looking at the structures of the trees, we can make some guesses about demographies of these populations, etc. But like I said, that it's just to give you an idea as to how we use contemporary DNA sequences in order to draw inferences on our evolutionary past. This is the kind of work that we do. Obviously, I am dealing with DNA sequences, and these DNA sequences belong to the domain of big data. So, much of the algorithms, etc., that are used are also used here, but essentially ultimately it's probabilistic frameworks that are used. Okay, I will stop there with so much of paradigms and methods. So, like I said, that we came out of Africa and we saw that we were not alone. We met at least 2 other species that we know of. One is the Neanderthals, and we came out and moved around in the Eurasian landmass, and that's where we met the Neanderthals. And we met another species called the Denisovans. There are lots of Neanderthal fossils that have been found now. Many of them have actually been sequenced. The DNA sequences of many Neanderthals are known. The Denisovans lived in a cave, and what was found was a particular finger bone in this cave, and the DNA was sequenced. Initially, the thought was that they were probably Neanderthals, but when the DNA sequences were compared, they were so different. There were so many differences between the Neanderthal DNA sequence and the DNA sequence that was the DNA that was extracted from this finger bone and sequence. There were so many differences that it could not belong to the same species. So this was called Denisovans because this cave in Siberia is known as the Denisova cave. Whoever lived there was called a Denisovan. Now there are more remains of Denisovans that have been found, and we have multiple DNA sequences of Denisovans, not as many as Neanderthals, but still multiple DNA sequences. So Neanderthals are now recognized as a separate hominin group. They existed for almost 500,000 years in Europe and Western Africa. Again, these time estimates have been done from DNA sequences. They roamed in Eurasia, living in small groups, and again, like I said, demographic features also can be estimated. So there were probably between 20,000 and 50,000 individuals. There was a disappearance of Neanderthals between 30 and 40,000 years ago, and at a time when the humans spread in large numbers outside of Africa and the Near East. So this was an enigma. Why did the Neanderthals vanish? Their history is unknown. Their relation was unknown. Their relationship to modern humans was a source of speculation, fascination, and debate. And there were 2 major theories regarding their vanishing, Neanderthals vanishing. One is we killed them and exterminated them, and that was a favorite hypothesis. But people who opposed that hypothesis also said that Neanderthals were also stone users. So if we were trying to kill them using stones, they could also kill us because they were also stone users. But anyway, that was one dominant hypothesis. The second hypothesis was that Neanderthals are direct ancestors of the present day Europeans. So we will see these 2 hypotheses have been tested, and as a matter of fact, the test of that hypothesis resulted in a Nobel Prize in 2022. This is Svante Pääbo, whose discoveries led to concerning the and this is what the Nobel citation says, that he was awarded the Nobel Prize for his discoveries concerning the genomes of extinct humans and human evolution, which is really true. Extinct humans is what he was. But for those of us who know this kind of work, firmly believe that it was not just for sequencing the genome of ancient humans or hominids that he was awarded the Nobel Prize for. He actually provided all of the methodology, and the methodology was not simple. There was this fragmentation of DNA, there are changes in the DNA bases, there are blocking lesions that actually prevent purification and amplification. There are a whole lot of things happen in ancient DNA. They are all damaged DNA. There is contamination with exogenous DNA, lots of bacterial contamination, and so on and so forth. And what Svante was able to do is essentially one after another get rid of all of these problems, and today even in our hands ancient DNA sequencing has become completely easy. So many people, there are many ancient DNA labs throughout the world. For those of us who have known him, who have read him, who have heard him, we know the kind of frustration he went through when one after another the various kinds of techniques failed and he was unable to sequence the DNA, but eventually he succeeded. So it's our belief that the success or the Nobel award was not just for the sequencing of the DNA, but for elucidation or discovery of these various methods that has resulted in our ability to sequence the DNA. He was the first to sequence the complete genome of a Neanderthal from the Altai Mountains, and he was also the first person to sequence the Denisovan, and comparison of these 2 actually resulted in the discovery of this new species called the Denisovan. They have a evolutionary history which is distinct from that of the Neanderthals. The common ancestor of Neanderthals and Denisovans lived somewhere in Africa 500,000 years ago. Like I said, that these individuals whom we met after we came out of Africa also evolved in Africa, and they spread out of Africa. And then, so what happened to the Neanderthals? Modern humans arose in Africa about 150,000 years ago, 130,000 years ago. We started moving out of Africa about 100,000 years ago, maybe 80,000 years ago. We reached the Near East. That's when we reached the Near East before we met the Neanderthals, and we started mixing with the Neanderthals. Rather than becoming their enemies, we actually started mixing with the Neanderthals. We also mixed with the Denisovans who lived in Southeast Asia. And this mixing was actually mixing of genomes, meaning that we interbred with them. We interbred both with the Neanderthals and with the Denisovans. And how do we know we interbred? There are pieces of Neanderthal genome in the human DNA even today. Each one of us would probably have a little bit of Neanderthal genome and a little bit of Denisovan genome. And I'll show you what percent in a moment that we discovered not so long ago. So the children born out of these Neanderthal human matings or Denisovan human matings, they were absorbed in the human group. So all the children of these matings were absorbed in the human group. So over a period of time, the demographic size of the Neanderthals and the demographic size of the Denisovans, they started to decline because the children didn't go with the Neanderthal parent or the Denisovan parent, they came with the human parent. So essentially, we absorbed the Neanderthals and the Denisovans into our own gene pool. That probably is the reason for their extinction. But like I said, that they live on. Both the Denisovans and the Neanderthals actually live on, because 1 to 2% of the genomes of modern humans belong to the Neanderthals or the Denisovans or the Denisovans. Up to 7% of the genomes of these people in Melanesia belong to these two ancient hominids. So this is again, these are data that's been analyzed, data from where admixture levels have been estimated using genomic data. So we now know that Denisovans and Neanderthals contributed some amount of DNA to the modern humans, and the Neanderthals and the Denisovans also mated, and they contributed genomes to each other. But the interesting part is that as we analyze this data, our own data and data analyzed by others, we found that there are DNA sequences in the humans that neither belong to the Denisovans nor to the Neanderthals. So they probably were some other hominins who were our ancestors with whom we interacted, and they also provided their DNA to us. There is now more and more evidence that's coming up, and higher amount of DNA in our genomes that belongs to these potential ancestors. So when we looked at the human DNA, and when we meaning I, my group, my collaborators, and so on, when we looked at the human DNA, what we found was that there were these little pieces of human DNA, little pieces of Neanderthal and Denisovan DNA. That was not surprising at all. Then we found among the Jarwas and the Onges of Andaman Islands some little pieces of these yellow fragments that neither belong to the Neanderthals nor to the Denisovans, and didn't really, when we aligned them to the human sequence, didn't align. So we looked at multiple population groups of mainland India, and we didn't find these yellow bits in pieces of in these populations. They were only found in the Jarwas and the Onges on whom we had DNA sequences. Eventually, what we did was to ask ourselves, what is the time to the most recent common ancestor of these DNA sequences? They went back to the time of the when Neanderthals and Denisovans were alive. So these unknown DNA pieces belong to an ancestor who were neither Neanderthals nor Denisovans, but lived around the same time. That's all we did. We also incidentally found these yellow pieces in some populations of Southeast Asia. So we started our collaborations with Southeast Asia, and we found these yellow pieces in some populations in Southeast Asia. We published our paper, and essentially we could provide no further evidence as to who that unknown hominin may have been. While we were doing our study, at the same point of time, there was another group of people who were looking at genomes of Melanesians, and they also found that they also found evidence of this mysterious hominid that was neither Neanderthal nor Denisovans. So, this still remains it still remains a hypothesis as to who that particular mysterious hominid may have been. We have not been able to find, or at least archaeologists haven't identified another species using fossil remains, etc. But genomic data almost unequivocally shows that there was at least one species of mysterious, one species of hominids who have not been discovered. We are unusual animals. We have a wide geographical distribution, and we have adapted ourselves to a wide variety of environmental conditions. However, we choose mates from within our own group, however we decide to use that word group. As a result of our tending to mate within our own groups, genetic variations tend to remain localized within groups, and groups also remain some measure of genetic distinctiveness. Like I showed you in that phylogenetic tree, there will be some populations that will have distinctive characteristics, and those are hallmarks of those populations. And those are also used to estimate admixtures between populations. And the barriers to admixture are geographical, physical barriers. So, for example, if there are two populations living on two sides of the Himalayan mountain belt, chances are very slim that they are going to admix, that there is going to be exchange of mates between the two populations. So, geographical barrier is a major barrier to admixture. There are also cultural barriers. If you assume that language is a part of culture, culture and geography are two major barriers to human evolution. Well, the expectation in terms of human evolution is that if you look at genomic diversity of contemporary populations, if you look at populations that have a huge amount of genetic diversity, the inference is that that population must have evolved for a longer period of time, because mutations accumulate over evolutionary time, and therefore, mutations also increase genomic diversity, and therefore, if you have populations that have a huge amount of diversity, the inference is that they are older than other populations that have lesser genomic diversity. So, if you look at the global scale, Africa has the highest amount of genomic diversity, followed by Asia, and then followed by the other continents or subcontinents. And therefore, this is consistent with the view that we evolved in Africa, and therefore, our evolution, the populations of Africa have evolved for a much longer period of time than populations of other continents or subcontinents, including Asia. We have other ways of looking at it. So, this is Y chromosomal DNA. This is essentially Y chromosomal chromosomes, as you know, are transmitted by the father to all his sons. This is Y chromosomal DNA, and the way that it has been arranged, these are signatures of the Y chromosome, specific signatures on the Y chromosome, and the way that it has been arranged is that the ones on the left are the oldest signatures and the ones on the right are the newest signatures. These signatures of course are defined by a constellation of mutations that are present in those segments of the Y chromosome. If we look at the worldwide distribution, so each pi essentially represents different kinds of signatures of the Y chromosome on a sample of Y chromosome that has been drawn from that particular area or from that particular population. What you see is that the newest Y chromosomes are here. These newest Y chromosomes are like Q and so on. So, those are in the New World. The oldest Y chromosomes are Y chromosomal signatures are in Africa, like for example these colors that you see, they are in Africa, and of course the middle-aged signatures are all in Asia. Asia also has a great diversity of Y chromosomes. So, this is again consistent with the view that we evolved in Africa, and therefore our oldest Y chromosomal signatures are in Africa. We moved out of Africa, across the Bering Strait, and populated the New World, and that was not so long ago, maybe about 25,000 years ago. So, that is where the newest signatures arise. This is Y chromosome. I'll talk about mitochondria. Mitochondria is, I have my mitochondria that's been donated by my mother, transmitted by my mother. My sister also has mitochondria that also has come from my mother. So, essentially the mitochondrial DNA signatures track female lineages, and again these signatures have been named differently, but essentially it has been arranged from left to right, oldest to the newest. And again, if you look at the distribution of female lineages, it is exactly the same as the features are exactly the same as the features of the Y chromosomal lineages, which means that the oldest are in Africa, the newest are in the New World, and all of the middle-aged signatures are in Asia and Europe. Again, consistent with the view that we evolved in Africa and moved out of Africa to populate other parts of the world. we moved out of Africa to populate other parts of the world. I was most interested in looking at human evolution in India. Over a period of time, expanded my horizons to look at human evolution in Asia. We collaborated with a large number of population geneticists, and essentially what we find is that these are, you don't have to read any names, these are names of population groups from different parts of Asia. I will draw your attention to only one particular region of this particular phylogenetic tree. So, this is the phylogenetic tree providing the relationships among these populations. This is the root of the tree. The tree is rooted using an outgroup of Africans, and that is how we usually root trees. So, that is the root of the tree, and if you look at this particular root, which is one of the oldest roots of the tree, and if you look at the branches of that particular tree, all of the branches belong to India. So, essentially there are not too many groups, there are like 12 groups from India that we sample and analyze, and this is using not yet whole genome sequences, this is using 1 million chips. So, essentially 1 million points on the genome that are variable across individuals, that is used as a microarray. We use those microarrays in order to identify variability among individuals belong to different populations, and essentially what you find is that there are two groups, one group of populations that come from North India, another group of populations from East India, and they are different from North and East India and South India, which turn out to be different, but the major point that I want to make is that one of the oldest populations belongs. We root ourselves in one of the oldest roots that came out of the process of human evolution. So, after humankind came out of Africa, one of the earliest trails of migration was into India and then to other parts of the world. So, in some ways we are fairly ancient in terms of human evolution. If we look at our ethnic composition, we have a large number of tribal groups, they have simple social organization, they speak languages belonging to these language families. We have a large number of castes, including subcastes, they are about 4,000. These numbers do not come from me, these numbers have been estimated by the Anthropological Survey of India. They have a complex hierarchical social organization and carry out a variety of occupation. They speak these different languages, and then we have religious and migrant groups, and they also have similar complex social organization. They do a variety of occupations. The tribal populations usually do simple occupations, hunting and gathering primarily, but today farming as well, and they speak languages that belong to these three language groups. You may have noticed that the Austroasiatic language group is spoken only or exclusively by the tribals in India. I will describe some data that we used, about 600 individuals drawn from about 42 ethnic groups. So, roughly about 10 or 15 individuals per group, that is all we could afford at that time. We generated whole genome sequence data, we also used a 1 million SNP chip or SNP array, and I will describe some of this in a minute. We eventually, this is only India, it was a larger study called Genome Asia study, and I am not going to present the Asian data, just going to present some features of the Indian data that belong to this larger project. Before I actually present this, I also want to mention that there is a huge project that is going on in India right now, and that is called the Genome India project, funded by the Department of Biotechnology, where 10,000 individuals, I am only going to present data on 600, 10,000 individuals whole genome sequences are being done. The data are right now in the process of being analyzed. It is anchored in the Center for Brain Research. They are spearheading this study in the Indian Institute of Science, and you will hear snippets of this particular project in the next talk. We use these data, the data on the 600 individuals, to estimate Neanderthal admixture. And what we estimated is that, well, first of all, there is some variation among these population groups. Again, don't worry about the population groups. There is no geographical pattern of admixture, so the proportions of admixture don't follow a particular pattern. The amount of admixture is between 2% and 3%. That's Neanderthal admixture. Denisovan admixture is even lower, about 0.1 to 0.5%, and again there is some variation across populations about the extent of Denisovan admixture. Interestingly enough, the Denisovan admixture shows some patterning. The tribal populations have the highest amount of Denisovan admixture, followed by the caste, followed by the upper caste, and followed by the Pakistanis. The Pakistanis have, we've also, this was a Genome Asia study, so we had access to some data from populations in Pakistan. They had much lower amount of Denisovan ancestry. So this is also quite interesting. Again, we don't have really a neat explanation of all of this data. Some hand waving arguments we did make in our paper that was published in Nature, but essentially it's not really convincing as of now. The next question, or an earlier question that we had asked is, okay, so we have a huge amount of diversity among our ethnic populations. Where did this diversity come from? Are there multiple ancestors that we descended from? Even if we cannot identify who those ancestors are, there are now methods of estimating how many ancestral populations there may have been with whom we have admixed, and the extent of admixture with those hypothetical ancestral populations. There are now statistical methods of doing it. Not getting into the ways of doing that, but the first thing that we noticed, and again this uses a 1 million SNP chip data, and essentially the first point that came out after, as we started analyzing the data, is that Andaman and Nicobar Islanders, so these are based on those 1 million data points, we extracted principal components, and this is the first principal component and this is the second principal component. What we find is that these each dot represents an individual, so these individuals stand out. Where do these individuals come from? They are all individuals who have been sampled from the Andaman and Nicobar Islands. So the islanders stand out, and each of these individuals are from mainland India, and they are fairly distinct. The islanders are fairly distinct from the mainlanders. If we now exclude the islanders and just try to analyze data on the mainland individuals, what do we find? We find that there is some amount of population substructuring. These individuals are similar and quite different from these individuals, quite different from these individuals, etc. So there is some amount of population substructuring that we find. And those are the 4 major clusters that we can find, and these 4 major clusters actually come from different geographical regions. So this is essentially north and east geographical region. These are individuals from southern India. These are individuals from central India, the Gonds primarily, and so on. So there is, along with the genomic substructuring, we can relate genomic substructuring to the geographical affinities of these people from where they have been drawn. We estimated admixture using these methods that I am not describing. So first of all, let me tell you that on this axis, on the x-axis, there are thousands of dots which you can't distinguish. Each dot represents an individual. So what we are asking is, if I take an individual's genome, does that genome belong to one single common ancestor or multiple common ancestors? So these ancestors are denoted by different colors: the green, the red, the blue, and the teal, and so on and so forth. The y-axis represents this starts at 0 and this ends at 1. So this is the proportion of admixture. So if you take an individual here, this individual's genome comprises a mixture of 4 different kinds of ancestors, because there are 4 different colors that are you can see on this vertical line, and that belongs to a single ancestor, single individual. And the colors are not all of the same height, which essentially means that these 4 ancestors contributed differential amounts of their genomes to this particular individual. So anyway, that's what this is. And if we look at, and so what we can see is that there is a huge amount of variation in admixture estimates from these 4 hypothetical ancestors. There are 4 colors, so there are 4 hypothetical ancestors. The extent of admixture is quite different from for these different individuals. And if you look at the clustering of these individuals by their admixture levels with the hypothetical ancestors, what one finds is that these individuals have been derived from a common ancestor which is common to the Indo-European speakers. These are from the Dravidian speakers, these are of the Austroasiatic speakers, and these are from the Tibeto-Burman speakers. So, essentially our genomes, contemporary genomes of Indians, are a mixture of 4 ancestral genomes, and these ancestral genomes cannot be identified with any particular population today. Those are inferred ancestors, but the maximum likelihood estimate of those number of ancestors is 4. We published this in PNA some years ago. So, I will turn to the last bit of my story, essentially to look at cultures and see how genes and cultures co-evolve. How do we define culture? Culture is information that is capable of affecting behavior of individuals which we acquire or learn from other individuals, and cultural change can be modeled as a Darwinian process, and I am not going to get into the modeling of cultural change as a Darwinian process. Essentially, culture can be, or whatever we learn as a part of culture, can happen in one of 2 ways. One is by word of mouth. I can hear something and adopt that particular, or learn that by hearing of that particular trait from somebody else, or somebody can actually physically move to my house and teach me how to do it, right. So, there are these 2 models. One is called cultural diffusion, where there is diffusion of ideas with no physical movement of people, and the other is called demic diffusion, where there is diffusion of ideas through actual movement of people. How do we distinguish these 2? We can use genomic data in order to distinguish these 2, because with demic diffusion there will be genes that will move with these people. On the other hand, with cultural diffusion there will be no physical movement of people, and therefore genes will not move. So, we can use genomic data in order to distinguish between demic diffusion and cultural diffusion. Let us look at one aspect of our culture, which is farming. Farming arose in the Fertile Crescent region, and this is a particular genomic signature that has its highest frequency in the Fertile Crescent region, which is Syria, Lebanon, Turkey. From various kinds of fossil evidence, people now know, fossil hunters have told us that agriculture in its modern form arose in the Fertile Crescent region. We as genome scientists also have been able to identify a genetic signature that dates back to around that same time where archaeologists told us when farming arose about 10,000 years ago, and this genomic signature also has its highest peak in the Fertile Crescent region and dates back to about that time. As you move away from the Fertile Crescent region, you see the frequency of this signature declining. This is consistent, and this is how agriculture also has spread. So, this is consistent with demic diffusion of agriculture if we consider farming practice as a cultural phenomenon, where you are teaching somebody else how to do farming, and this is consistent with demic diffusion as opposed to cultural diffusion. If it was cultural diffusion, you would find patches of these or you would find discontinuities of this particular signature, but what we find is that from the dark shades mean that the frequency of that signature is the highest, and as the shades become lighter, it essentially means that the frequency of that signature is declining. However, there is no discontinuity. We see that it is continuous, and therefore, this is consistent with agriculture or farming practices being taught by people who actually move to another place and teach people there how to do farming. So, this is what we believe farming, this is how we believe farming has spread. Farming also came into India, and therefore, we wanted to test whether the same phenomenon also holds as far away as in India, and we refined this genetic signature using various other kinds of techniques. We published this and so on, but anyways, this is what our data have told us, that the colors have changed. So, this is the Fertile Crescent region, and as you can see that there is decline in frequency, not only away from the Fertile Crescent region, but as we come into India there is continuity, and therefore, farming in its modern form that arose in Fertile Crescent region came into India through a process of demic diffusion. You can also see that there are other smaller islands where something else has happened slightly different, but I will not come to those differences. Those are seats of early agriculture that had taken place in India, but that were invented in India, early agricultural practices that were invented in India, but Unfortunately, it did not spread to other parts of the world very much. It remained localized. There is one in South India, which is not shown here very clearly. There is one in the northeast of India, but I am not getting into that. So, this is from Jared Diamond's work. Essentially, he collated data from various sources and told us that this is the Fertile Crescent region where agriculture arose about 10,000, 11,000 years ago, spread to other parts of the world, and there were also rice, for example, arose about 9,000 years ago. Rice farming arose in China. This is primarily wheat and barley, and then there were other kinds of cereals that were along with farming. So, farming essentially was a demic diffusion process. Simultaneously, concurrently with farming, there was also domestication of animals. And the domestication of animals, if we leave out the dog, the domestication of animals of goat, sheep, cattle, and so on and so forth, coincide with the invention of farming practices. So, the domestication and agriculture, they went together, and it was of interest for us to find out certain key features of domestication process as well. I will skip this. And again, the spread of domestication of cattle, sheep, etc., happened in this particular way. Won't get into the details of this. The one point that I would like to mention to you, which is again a part of culture, part of biology, etc., as all of us know that most babies can digest milk without any problems. But many of us, I do not know whether most of us, but many of us cannot actually digest milk. And after the weaning phase, the reason why the babies can digest milk is that they have a particular enzyme called lactase, which converts lactose to other forms of sugar. They have this particular enzyme called lactase, which is produced by a gene, and that gene gets shut off after the weaning phase. But then today there are many people who also, many adults who can drink milk. How did this happen? And interestingly, it happened with domestication of animals, domestication of these animals. So, technically it is called lactase persistence. Lactase does not persist, it gets shut off, but there is an allele that allows lactase to persist, and this has spread from high frequencies in less than 9,000 years from the inception of farming. So, farming and domestication went together, and this particular gene underwent some change, and lactase persists, the enzyme lactase persists, and therefore, we are able to digest milk. Many of us are able to digest. I can't, but many of you probably can. So, this is the distribution of the frequency of digesters, people who can digest milk. You do not have to get into the gory details of it, but as you can see that there are differences. These are the color frequencies. So, there are differences across the world in the frequency of digesters. Today, 35% of the global population, mostly people with European ancestry, can actually digest milk even as adults without a hitch. If you look at more continuous distribution of these, this is how the continuous distribution looks like. The lactase persistence has a genetic basis and follows a dominant pattern of inheritance. In other words, it is autosomal. There are two genes. If you have one particular gene that has an allele that allows lactase to persist, then you will become a digester. And the lactase non-persistent is a recessive phenomenon. The lactase gene, this is the lactase gene, that particular gene produces an enzyme that helps digest lactose. But the interesting part is that the shutting off of that gene does not happen from within the gene. It happens from within an adjacent gene that has lots of that interacts with the transcription factors of this particular gene and shuts off this gene. So, this is a cell cycle regulatory gene. Exon 17 of this gene plays a critical role as a regulatory enhancer regulation of this lactase gene. I will skip this. There are multiple transcription factors. These are lactase non-producers, which is the normal thing. There are certain or certain mutations that take place. These mutations enable the lactase to be produced continuously, and therefore, these are lactase persisters. So, we can look at these specific single nucleotide polymorphisms or mutations and correlate that with lactase persistence. So, there are multiple mutations that are associated. Some of these are widely studied and associated with lactase persistence. Some of them are incompletely studied and not very well known. Some of them are fairly rare. So, if you look at a specific mutation, this is the most prominent mutation. This is one particular mutation that changes C to a T at a specific position in that MCM6 gene. Enables lactase persistence, and if you look, this is a T allele that increases lactose expression by creating a new binding site for a transcription factor that interacts with the human HNF alpha to bind to the lactase promoter. So, essentially this kind of activity, the binding of the transcription factors promoted by that particular mutation enables lactase persistence, and this is what the frequency of this mutation, the T mutation, is in various parts of the world. And therefore, as you can see that the T mutation is very frequent in Europe, so Europeans can easily digest milk. In parts of Africa, it is almost absent and people don't digest milk. So, what do we know? We know now that we have Neanderthal sequence and we have the human sequence both from digesters and non-digesters, we know that Neanderthals carried the ancestral allele. And it is also known that early farmers were lactose intolerant. So, they also did not have the persistence allele. This persistent allele is estimated to have arisen between 20,000 and 9,000 years ago. As you can see that this is a very broad, again lack of data, and that is the reason why it is very broad. But essentially around the same time when agriculture arose, domestication arose, and the T variant also arose at the same point of time. And it spread rapidly and became common about 5,000 years ago. The rapid spreading is because of positive selection. Individuals who are able to digest milk were able to derive a lot of nutrition from milk, and that is the reason why it was positively selected. We have genomic evidence now that it was positively selected. This is the specific core region which led to that C to T mutation, and once that mutation took place, if these individuals had the added advantage of digesting mutations and ability to survive longer without getting diarrhea, then it actually carries a whole set of correlated changes, correlated sequences around that particular mutation and goes on without recombination, and over a period of time recombinations will arise, etc. So, essentially what you find is that you have these large chunks that actually remain exactly the same across individuals if selection happens. So, these are core haplotypes with the LCT non-persistent, with the lactase non-persistent allele, and these, as you can see, is small. But once that mutation arose, you find large chunks of DNA being carried along with that particular T mutation that leads to persistence, and therefore, this is an evidence of positive selection happening because of this large chunks of DNA that goes along with the core mutation. Again, I am not going to get into the details of it. The question was, is it demic versus cultural diffusion? People who were herding, people who were domesticated, they were teaching the art of domestication to other people, and this art of domestication was correlated with our ability to digest milk and so on and so forth. So, was this demic versus cultural diffusion? So, that was the hypothesis that was being tested, and eventually what transpired by analysis of these data is that it was not versus, it was both. These yellow parts are the parts where demic diffusion took place, and the red parts are where cultural diffusion took place. So, essentially it was a combination of both. Unlike agriculture, where people had to be taught how to do the farming, some of this had actually spread by word of mouth. So, it was there was a combination of cultural diffusion and demic diffusion in the spread of domestication, and therefore, the spread of this particular allele that leads to lactose tolerance. I am not going to get into this. I think I am overshooting my time. There is an interesting hypothesis that has led to or explanation of the positive selection. I will not get into it. So, essentially to sort of end my talk, essentially in human history, we can look at genes in isolation and reconstruct human evolution, but when we want to look at culture and genes, what we find is that there are plenty of examples. I have given you two. Two examples, one of agriculture, one of another associated cultural trait of domestication that also impinges on human biology in our ability to digest milk. So genes and cultural traits are often co-evolve. So in human history, they often co-evolved. This is the end of my talk, but I have one more slide, and which I think is my most important slide, and I actually wish to, I know that you can read that slide, but I wish to read it out along with you. This is from Maya Angelou, who wrote this, who's a poet, an African-American poet who wrote this book called Wouldn't Take Nothing for My Journey Now. So she says that it is time for preachers, the rabbis, the priests and pundits, and the professors to believe in the awesome wonder of diversity so that they can teach those who follow them. It's time for parents to teach young people early on that in diversity there is beauty and there is strength. We all should know that diversity makes for a rich tapestry, and we must understand that all the threads of the tapestry are equal in value, no matter their color, equal in importance, no matter their texture. Thank you very much.
Thank you, Professor Majumder. We have time for a few questions since we are running a little bit late.
Hi, fascinating talks, really amazing. A lot of questions arise in terms of thinking about how we evolve, right? I mean, the fact that we interbred with Denisovans and Neanderthals is interesting, but you describe this process where we essentially assimilated them, right? What is this a function of? Is it a function of population size? I mean, you described the Neanderthals.
You're talking about Neanderthals and Denisovans?
Yeah, with humans, right? Was it a function that Neanderthals were relatively small in population size compared to?
Because their offspring, matings with humans, the offspring were going with the humans and not with the Neanderthals.
Why would that happen, though?
Why would that happen? Again, I think it's a cultural phenomenon. Probably humans were able to provide more tender, loving care to the babies. Why would that happen is a $1 million question. None of us will have an answer.
But you think the fact that we interpret rules out other possibilities, like there's a disease that came that wiped out the Neanderthals, or there was actually, some wars which gradually reduced their population.
If there was a disease that wiped out, then that was also a hypothesis that was done. We would find the impact of that on the genomes. There will be sudden decline of the genomes. Again, in terms of where we look at haplotypes, etc., one can actually figure out whether there was a sudden demographic decline, and that did not happen. It was a slow process over a long period of time. Again, if the demographic size of the Neanderthals to begin with was larger, it would have taken much longer time. So again, the point is that you set up hypothesis, you get data to shoot some reject your null hypothesis, and then you come up with the best possible explanation. So this is the best possible explanation as of now. Other explanations are of course possible, but we don't have data supporting other explanations.
Thank you. We will take only a few questions. Yes.
Sure. Hi.
Is there data to ascertain at the moment whether the mysterious hominid that shows admixtures was more ancient or less so?
No,
with respect to
we actually estimated the time.
fluorescences. No,
they all go back to the Neanderthal Denisovan time frame.
I see. So that it marks the Denisovan.
Yes, yes. So that was one of the initial questions. So we looked at those two DNA bits and estimated the most common ancestor, time to the most common ancestor of those DNA bits. They came back to that same Neanderthal ancestor time frame.
I see. And any idea as to why the fluorescences does not show much of an admixture, possibly because of migrational constraints?
The flores, they are an outlier, right? They were short. I know nothing about hobbits, and by the way, the sequencing has been done of the hobbits, so we don't even know.
So it's archaeological more than genetic.
It's archaeological, yes.
So this will be the last question. Who wanted?
How easy is it to sequence ancient DNA from tropical regions? Because if I understand correctly, most of these are from...
Absolutely impossible. I mean, people have been trying. We've been also tried. I've given up. It's so contaminated, so fragmented. It's just absolutely impossible to isolate human DNA from the sequence that we derived. It's very, very difficult. So much of fungal DNA, bacterial DNA, it's just horrendous.
Okay, so and we're running late. You can always ask the question.
Sure.
So I'll take the last question, take the privilege. So New Guinea or Papua New Guinea, has the highest, genetic diversity, also probably language and culture. So what's the
Population sizes are very, very small.
I see.
The genetic drift is probably, I don't want to explain what genetic drift is, but this actually changes allele frequency. Genetic drift changes allele frequencies without selection. Essentially, it's a random process. So when population sizes are small, the frequencies of alleles can change rapidly without any selection happening, and that's just a random process called genetic drift. In populations of Papua New Guinea, where they are isolated populations, this is probably the major reason why there are so much of allelic differences between the populations.
And how about the culture and the language? Because that's also very diverse.
I don't know that very well, but I
There
don't
are
know how
800
that means
languages.
that they were admixed by multiple sources, right? I would imagine, because language doesn't evolve de novo as rapidly. Once they appear, they evolve faster than genes. The mutation rates of language is faster than the mutation rates of genes, but it's essentially because of admixture, I would imagine. So I don't know about the language, but I know that their diversity is very high, and the explanation of that is because their population sizes are small, so genetic drift is at play.
So apparently Papua New Guinea has the highest number of languages, in a very small region that they inhabit. So this is something I also learned very recently.
I look up also.
Thank you so much. Thank you
Thank
so
you.
much,
Thank you.
Professor Majumder.
Thank you very much.
So we have a coffee break now. We'll come back at 10:35.